Namaste students, myself Dr. JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. So today we will discuss the topic, the filtration and microfiltration. So this topic comes under the subject upstream and downstream process. So the filtration, a little bit we have already discussed in the previous lectures. But we will discuss in detail what is the filtration and the microfiltration processes. So the filtration is principally it is a physical separation method. So you use the physical membranes or uh, things uh, uh, having the specific pore sizes, which is used to filter out the insolubles from the liquid broth or the water or the media, etc. So the extent of the filtration or purity of the product uh, is determined by the size of the pores in the membrane. So when you decreases the size of the membrane, pore size of the membrane, you will get the more purer product. Is the larger particles will not allow to pass through the specific size of the pore sizes membranes. So the larger are fully removed. Suppose if you have the membrane of a specific pore size, so the particles which are greater than that specific size will definitely removed, fully removed, and the smaller particles, the smaller from the size of the membrane, pore size of the membrane, will also be inhibited by various methods, like if the membrane is double membrane is there, so the the channels of the pores are the interlocked by the other layer of the membrane. Okay, or it may be blocked by creating the filter cake. Okay, so the smaller particles may also be removed, but not fully. Partially they are removed, but definitely the larger molecules will fully removed from the broth media or the water or the culture media, etc. So there are various types of the filtration processes, various types of the filters are there. One is the depth filter. So it is made up of the glass wool, asbestos, or the filter paper. So the particles are trapped within the matrix and the fluid passes out. Okay. So the another is the absolute filters. So the filters are with specific pore sizes that are smaller uh, than the particles to be removed. So what is the difference between the depth filter and the absolute filter? So as name suggests, the absolute means the specific, absolutely fixed size of the pore. So this is present in the absolute filters. The pore sizes are absolutely determined that what a specific size of the pores are there. But in the depth filters, it is a glass wool material or the asbestos or the filter paper. So these the pore sizes of the depth filter are not absolutely correct, or it may vary. So the absolute filter is used generally for isolating the or separating the bacteria from the broth culture or culture medium. So next is the rotatory drum filters, rotatory drum vacuum filter. So it is a specific type of the filter where membrane or the filters are made in a drum-like structure. So frequently, it is frequently used for separation of broth containing the 10 to 14% solids and particles in the size of the zero point 0.5 to 10 micrometer. The rotatory drum vacuum filters have been successfully used for filtration of yeast cells and the filamentous fungi. So this is the specific purpose or the application of the rotatory drum filter. So the filtration unit consists of a rotating drum partially immersed in a tank of broth. So here is a rotating drum is used, which is partially immersed, partially dipped in a tank of the broth. Okay, so as the drum rotates, it picks up the biomass which gets deposited as a cake on the drum surface. So this filter cake can be easily removed. So suppose a drum-like structure is immersed in submerged in a broth medium, and the drum is made up of a filter. So when it rotates, it will take the broth, and while rotating, the media is simultaneously uh, filtered filtered, and the insoluble materials are uh, deposited on the filter, or on the drum as a filter cake, and which is easily removed. So next is the membrane filters. So membranes filters are the specific membranes with the specific pore sizes. So the membrane filters have a problem of the clogging. So there are two types of membrane filtration: static filtration and the cross flow filtration. So in the cross flow filtration, the culture growth is pumped in a crosswise fashion across the membrane. So this reduces the clogging process and hence better than the static filtration. So the microfiltration and the ultrafiltration will be covered in the next lecture. So we will stop here. So I hope you are satisfied with this session, and if you have any query, please write in the comment box. I will resolve your query. This session is powered by Digital Version 2, Jyoti Vidhyapeeth, Women's University, Jaipur. Thank you.